All right. So for today's lecture, we're going to be focusing on protein biochemistry. What that means is we're actually going to be focusing on a few different approaches where we've spent a lot of lectures focusing on different. We first talked about proteins and amino acids, and we spent a lot of time talking about the different kinds of proteins that you see in cell and molecular biology. Now we're going to be focusing on how we actually try and measure and characterize those proteins in a research setting, as well as how that protein biochemistry is leveraged for really important methods of detecting certain proteins in the cases of rapid detection tests, uh, with the examples of COVID-19, a pregnancy test, and the anthrax test. Um, this is a cartoon that was created by a student a little while back. I say a little while back, but now I'm realizing that's much longer ago than I would care to admit. Um, but just trying to make a little light humor out of the importance of how these tests are actually, their specificity is important. Um, so the mechanism by which these tests work for a pregnancy test, an anthrax test, as well as a COVID-19 test, is lateral flow immunochromatography. And so what ends up happening is that you have some sort of biological sample. In the case of pregnancy test, it's urine. In the case of the COVID-19 test, a deep nasal swab. Uh, that are loaded into a test site, and there's a resin that flows, laterally flows the test sample across to where you'll be able to see detection of that protein of interest in the biological sample. Uh, the This is what's typically used clinically. In a research setting, there are a few different approaches to try and measure and characterize proteins. The uh, pre-lecture really went into in-depth description of a few of those, but we're going to be trying to focus on a few key particular ones today. And so, what I'm going to try and do a better job of is try and focus on like what the key take-home messages for the entire lecture, and then make sure that for each important slide, I'm going to try and communicate what the take-home message is. And it's okay if you want to raise your hand and say after a slide, what's the take-home message? Because if that's not communicated, or if you feel like that wasn't communicated, that's not on you, that's on me. So please feel free to jump in and ask what the take-home message is on a certain slide. Um, so one of the commonly used approaches for trying to characterize and measure proteins is electrophoresis. Does anyone want to help define for us what electrophoresis is? For those of you who might have been in Latin club in high school, you might be able to, or for those of you who've been working in a research lab already, you might be able to help describe what that is for us. And if you don't want to admit that you were in a Latin club, that's okay. So phoresis, the Latin word for phoresis is mobility. So what electrophoresis is moving a sample movement across an electric field. Um, and we actually use this movement across an electric field to be able to, in electrophoresis, to be able to separate molecules by their size and their charge. We're going to be talking about what that looks like in a little more detail in the next couple slides. But what ends up happening is that we apply a positive charge to the bottom of a gel, and the gel is in blue because I'm going to take a moment to describe what that gel is. Um, but the reason why we run a positive charge is because we want our sample that we load in this gel to run with the flow of current. Current is the flow of electrons. So I'll actually talk about what we do to try and match the charge in some cases of our proteins to have a negative charge so that they can flow with the current towards the positive end here. This gel is, when you think of a gel, you might think of a few different things might come to mind. But what I actually want you to think of is at a molecular level, these gels are a polymer network where there's a matrix almost. Um, whenever you hear polymer network, a good visualization can be try and envision the scaffolding that's on the side of buildings that are on construction sometimes. These just like this like network of beams crossing back and forth with each other. That's what this gel is at down to a molecular level. And so we can change the density of that polymer network, the density of that gel, to be able to control what proteins can run through this gel. And so what ends up happening is that we typically will have samples loaded on top of a gel, and we'll bombard them with negative charges in a few different ways. And what we'll end up doing is that we'll then run that current to separate these proteins based on their size. There are going to be some instances where we separate them based on charge as well, and we'll talk about that a little bit later in this lecture. But a common approach for electrophoresis is to apply a same charge to all of these cells so they can run through a current and be separated by their molecular weight or based on their size. When you run a gel like this, and after you run this, run your protein-rich sample, you'll have what we call a native gel. You just have gel with proteins embedded within this gel matrix, basically. You typically don't see bands here, like you typically don't see bands quite yet like this when you have a native gel, but this is just to help illustrate this separation. And you can employ a few different stains here to try and be able to visualize these proteins on this gel. So the take-home message here for electrophoresis is that it is moving of proteins across an electric field to separate them, in some cases by size and by charge. For a common electrophoresis approach that I'm going to talk about in the next slide, it's usually separating them by just their molecular weight. This most common form of gel electrophoresis is called SDS page. I'm going to break down the SDS and the page part and what that means. SDS is sodium dodecyl sulfate. It's this anionic detergent, anionic, negative charge. So that's one of the ways in which we bombard these proteins with negative charges so that they run across this electric field with the flow of the current. It also denatures. SDS is important because it does two things. One, it supplies the proteins with a negative charge. It also denatures them. When I say denature, and that denaturing is really important, we've talked several times about how biology exists in 3D. There's protein folding, but it can sometimes be hard to try and run a protein or be able to characterize a protein if it's in its folded up state. We want to unfold it. The unfolding of these proteins is denaturing. So when it's bombarded with a negative charge all across this protein, negative close to a negative repels. And so that bombarding with negative charge repels and promotes the denaturing of that protein. We also typically, it's not listed here, but we'll also usually heat denature a sample to try and further denature this protein so that all of the protein is accessible for downstream analysis. Finally, there's also, in some cases, disulfide bonds across a protein. You've seen that in the case of insulin. There's probably some other proteins that you guys have looked at for your projects that there's probably heavy disulfide bonds across it. Those are not going to be simply, those disulfide bonds are strong and won't simply be broken by heat denaturing or by sodium dodecyl sulfate. What ends up happening is that we need to use a reducing agent to try and break those disulfide bonds. A common one is called beta-mercaptoethanol, sometimes colloquially called beta-mercat. And so that reducing agent, again, this is a key point here. The reducing agent that we use here helps break those disulfide bonds. In any of the labs that you guys may have walked through or do a rotation in or shadow in, if you smell rotten fish, that is beta-mercaptoethanol. So you know that someone's probably about to do SDS page. Excuse me. So the page part just tells us what kind of gel is being used in this gel electrophoresis. It's polyacrylamide. It's a pretty standard gel that's used that is, in a lot of cases, people would have to make these gels usually, but now they're pretty cheap to go ahead and just purchase commercially. And so because of how affordable they are, and how tunable they are, polyacrylamide is a common gel that's used in gel electrophoresis. Excuse me. So again, we have bombarding of these proteins with SDS to try and create this negative charge on it. And typically, it's to try and help illustrate this bombarding, not something for you to memorize, but rather to help you understand. There's usually one SDS molecule for every two amino acids in a given protein. And so it's really, it is down to the amino acid level. It's really working to try and even in hydrophobic regions, and I'll show you in the next page how this works, even within hydrophobic regions, SDS is able to wedge in and provide a negative charge. It also promotes the denaturing. We can use a couple of other approaches to denature, heat denaturing, as well as reducing agents that break these disulfide bonds. And we use polyacrylamide to, as our gel here. And we're ultimately, after we heat denature SDS, use the SDS as buffer, we go from these proteins in these folded conformations to a just completely linear protein where every segment of the protein is now accessible and is now easier to run down a polyacrylamide gel. And again, with SDS page, I talked on the previous page where gel electrophoresis allows us to separate by charge and by mass. In SDS page, every protein is now negatively charged. So we're not separating by charge anymore. Here we are just separating them by their mass, by their molecular weight. So SDS has a bit of an aliphatic structure you can appreciate here, where there's a polar head. And so even within hydrophobic regions of a protein, this long hydrocarbon tail allows it to embed itself within even hydrophobic regions of a protein and provide this negative charge here. Polyacrylamide gels are usually consisted of acrylamide and bisacrylamide. And the polymerizing agents are ammonium persulfate, also colloquially referred to as APS, and TMED. And so what you end up having is just this network formed between acrylamide and bisacrylamide that creates this, again, scaffold-like structure that proteins are trying to be, our proteins are running through across this electric field. If you increase, again, you can think of this as, you can think of the scaffolding on the side of a building. If you increase the concentration of acrylamide or bisacrylamide, think of increasing the concentration of the bars and the scaffolding on the side of the building, you're going to have smaller pores. There's going to be a denser polymer network that leads to smaller pores. And smaller pores, only smaller proteins will be able to get through. So usually these gels will have oftentimes a gradient of concentrations where the larger proteins are caught up top and the smaller proteins make their way through this polymer network and make their way to the bottom of the gel usually. One thing that's interesting but not all that fun about acrylamide is it is quite dangerous. It's a neurotoxin and is carcinogenic. And so usually if it comes in a powder form, people have to be really careful because of how dangerous it can be handling it. But once it's in its gel form, then it's safe and stable. Unfortunately, all of us have been exposed to acrylamide, probably all of us, in some form or fashion. Acrylamide can be derived via a Maillard reaction. That's typically commonly found within high temperatures when starches are cooked. For example, if potatoes are cooked at a really high temperature, starches, the asparagine within starches can become acrylamide. So for example, French fries, there are trace amounts, some amount, I should say, of acrylamide found within potatoes. So, or within French fries, forgive me. So if any of you are eating French fries right now, I'm sorry. But yeah, so it's one instance of science meeting cooking there where there's actually a bit of a negative reaction when it comes to potatoes being cooked at high temperatures. So watch your acrylamide consumption. I'll stop there and give space for any questions. Yes. Mhm. No, so great question. So typically, these subunits will be separated during the denaturing step. And so when we talk about this separation, it's really just separating proteins based on their size. So for example, actually, it might be helpful if I go back one more slide. Let's stay close to the computer, forgive me. So in this bottom image here, what you can see is that we go from a mixture of proteins loaded into the gel at the top, and they are then separated by their size, where the larger proteins with a larger molecular weight kind of get caught in the network, the polymer network at the top, and it becomes denser and denser where, excuse me, the smaller proteins, those with a smaller molecular weight, get caught in the bottom. And so protein subunits can oftentimes be denatured and separated. In some other cases, proteins, you'll see multiple bands of a protein show. Trying to not jump too far ahead to what we'll discuss next lecture, but with methods where you're trying to detect a specific protein, and you'll see specific bands appear, you might see multiple bands appear if there's multiple subunits in some cases. But oftentimes you'll see just one single band if you're detecting one subunit of that protein, for example, because they'll oftentimes be separated out. But again, the key thing to note here is that we're separating by their mass, and these subunits will have different masses sometimes. For example, there are species of protein kinases. They have their regulatory domain and their catalytic domain. Those will be separated during this process, and they'll show up at different molecular weights, where typically at the top, a protein will be at 200 kilodaltons. I should actually take a step back and describe. So a dalton is the unit of molecular weight for a protein, grams per mole. And these proteins will typically be on the order of between, say, 20 to 200 kilodaltons. And so usually when you're doing SDS page, you reserve one lane where you have what's called just a standard ladder, where there's this mixture of very specific proteins that you buy, and they're at very specific molecular weights. And so you'll know this molecular weight is this band, this molecular weight is this band, this molecular weight is this band. And so you'll be able to determine and see where your protein is showing up, and if it's showing up at the right molecular weight, if it's at a certain kilodalton. And so typically it'll be somewhere between 20 to 200 kilodaltons when you're doing SDS page. Any other questions? Yes.
Why the charge is neutralized or like dissipated? Is it the high pH buffer, or is it something about the gel?
Yeah, it's SDS, that anionic buffer, where we are trying to make sure that our proteins run with the flow of current. And not all proteins have the same charge. And so we want to ensure that all proteins will run the gel the same, and that any separation of them is just due to their size, not anything to do with their charge. And so SDS ensures that all the proteins in some sample, whether it's a blood sample, for example, or something from a cell culture in a petri dish, that every protein is equally possesses the same negative charge per amino acid. And so that negative charge allows it to run with the flow of current when you're running a charge, an electrophoresis, a charge. You're trying to move across an electric field here. So the negative charge helps it run with the flow of current.
Points or like once it's denatured, that doesn't matter for how much SDS.
With how much SD, great question. Every protein has a different electrostatic equilibrium that is done away with when SDS is when every protein is in an SDS buffer. That's a great question. And we're actually, the last part of this lecture is actually going to be talking about the isoelectric point and how we kind of can leverage this to separate them by their charge. Did you have your hand up? Yeah, so if we want to run proteins down this gel, that can be encumbered with, in order to run them down this gel, we want to fully denature them, make them linear and not be in their folded up protein state, three-dimensional state, I should say. So to make them as linear as possible, sometimes proteins will have disulfide bonds form, but within a single protein. So we need to disrupt those disulfide bonds. We're not going to be able to do that with SDS. We're not going to be able to do that simply by heat denaturing them. So we need to use a reducing agent that disrupts those disulfide bonds to fully make these proteins linear. That's the role that reducing agents serve, and that's the reagent that I mentioned that smells like rotten fish, beta-mercaptoethanol. That's the most commonly used reducing agent in this case. But you don't have to memorize that. I would say the key thing to note is reducing agents help to disrupt and break these disulfide bonds. All right, we're going to go ahead and press on. But again, if any questions emerge, feel free to jump in. So we've run an SDS page. Now we have a native gel, a gel that just has all of our proteins separated out by their mass across this gel. You can use different stains now to actually directly stain this gel that will allow you to see what the total protein content looks like. So for example, one example of that is uh, a stain called Cipro Ruby, where you can just see where are all the stains given a specific protein ladder here in this first lane, where we know exactly what these molecular weights are. We can just stain for all the protein content and see what the total protein content is. If it's like a mixed bag of proteins, it can be really hard to try and you can't really distinguish what protein is what. However, if you're using, say, some engineered bacteria to produce a very specific protein, you can then use this approach to see, is it making our protein of interest at the specific molecular weight? The most commonly used total protein stain is Kamasi blue. And this is the structure for it. And it's actually derived from, it's a dye derived from blue gene fabrication. And so it's another Cipro Ruby and Kamasi blue, both stain for just total protein, all protein within the sample. ProQ Diamond and PhosTag actually specifically measure phosphorylated proteins. And so you'd be able to see within your given sample how much phosphorylated protein is there. And so the take-home message here is that there are ways to stain a native gel that give you information about either total protein or some subsection of proteins like phosphorylated proteins. It'll come back. Bear with me. All right. So we've talked about how SDS page enables us to separate proteins by their mass, by their molecular weight, by their size. You can think of molecular weight, size, mass as kind of interchangeable. But the key thing to note is that the units for molecular weight here are kilodaltons. And so you'll usually see kilodaltons indicated on the side of a gel when doing SDS page. If we want to separate these proteins based on their charge, there's actually this theoretical tube of a gradient of pHs is not just theoretical, but is actually used as a way to separate proteins based on their charge, knowing that, as someone described earlier, that proteins each have their own isoelectric point, their own pH at which they reach electrostatic equilibrium. You can actually load a sample in to a strip that has a gradient of pHs, run an electric field across it, and you'll be able to separate proteins based on their charge here. We're no longer separating based on size, but with a gradient of pHs, you're able to separate based on charge here. And so each protein will stop moving. They will no longer flow with the current once they reach their isoelectric point, and they're going to stop moving there. The protein, so with molecular size, there was a polymer network where proteins would stop moving based on their size because of the size of these pores, this polymer network. Here, they're going to stop moving across this gradient of pHs once they reach the pH that is their isoelectric point, because now they are at electrostatic equilibrium. They're therefore no longer going to have a net charge and are not going to move across an electric field. This method of separating proteins based on their charge is called isoelectric focusing. So again, we have a gradient of pHs in a gel. and a protein sample is loaded, and we have an electric field applied across this gradient of pHs. The proteins will migrate through until they reach their isoelectric point, the pH at which they reach electrostatic equilibrium. And so they're going to appear as a narrow band at that pH. And so they're because they've stopped moving there because they no longer have a charge. Now, what's interesting is that we can actually combine SDS page and isoelectric focusing into a combined approach to separate by charge and mass together. And this is actually called two-dimensional electrophoresis. The way this works is you'll first do isoelectric focusing, where you will run your sample across a gradient of pHs with an electric field. Once they all reach their isoelectric point, they'll stop moving. Now you'll take that gel and sit it on top of a polyacrylamide gel where you would separate by mass now, and you'll be able to separate now. They've been separated once you lay it across the top. Let's stay close to my computer, forgive me. Once you lay that isoelectric focusing gel on top of this other polyacrylamide gel, you can now see that they are separated laterally by their charge, and now they're going to move down and be separated by their mass. And so, excuse me. So the dimensions here is, horizontally, the x-axis would be being separated by charge, and the y-axis would be them being separated by their mass. And so this is why it's called a two-dimensional gel. What this actually looks like is this. And so what you end up having is these dots, and in some cases streaks that emerge. And what can end up happening is that these streaks are indicative of modifications to the same protein. There are some proteins that are phosphorylated. A phosphate group carries a negative charge with it. So for example, we talked about how kinases can add a phosphate group to certain proteins. In some of these cases of some of this blurring, this horizontal blurring, there might be instances of phosphorylation happening that lead to this blurring. There also might be, we talked about proteoglycans and glycoproteins. We have proteins that have some sugars that are added to them. Those sugars being added are called glycosylation. Some of this blurring might actually be a result of glycosylation events where proteins are modified and have sugars added to them. And so, excuse me. And so we have these smears on a 2D gel, largely because they have real biological relevance to how these proteins are modified. And again, horizontally, the units there would be the pH. Again, that's their isoelectric point. Vertically, the units there would be their molecular weight. It would be in kilodaltons. I'll stop here for a couple of questions. No questions? Yeah. Sorry. Yeah. So this is what it actually looks like. This is us running through like how it actually works, and what the result is this gel that has these dots and smears across it, and excuse me, the smears again are results of modifications to certain proteins. Could be a phosphorylation, could be glycosylation. Excuse me. But yeah, this is what a 2D gel looks like in real life. Yeah, so you typically won't have this labeling on it. You'll just have a gel uh, with uh, with these markings on them. But excuse me. And so what ends up happening is that you'll usually, well, I should say, let's go say this. If you stain this with, say, Kamasi blue or Cipro ruby, you'll be able to actually visually see the proteins this way. Otherwise, you won't visually see the proteins in the gel. You'll need to do something. And so the next part is trying, while the stains detect for total protein or all the phosphorylated proteins, the next step is how can we detect one specific protein? And so there'd be a way to label a specific protein. So say, for example, if we were interested in protein 21, we would, in this specific protein approach that we'll talk about shortly, you would actually see this smear of just that one protein for protein 21. But if you stain for Kamasi blue, you would see all of these light up. All right, so again, a couple of the approaches that we've talked about. We talked about gel electrophoresis at large. We talked about SDS page as a way to separate them by their mass specifically. Isoelectric focusing to separate them by their charge, and a 2D gel being able to separate by mass and charge together. If you have a gel now, that's either one of those approaches. If you stain with Kamasi blue, you'd see all the protein. If you stained with phosphate, you'd see all the phosphorylated protein. A lot of times we are interested in one protein, not all of the proteins. So the next approach that allows us to do that is called Western blotting. Some of you may have heard this before, also referred to as immunoblotting. It allows you to detect and measure one specific protein within a given sample. So Western blotting begins with SDS page. So we have SDS buffer. We have a polyacrylamide gel. We have our sample that's running across the gel and being separated based on its size. What's important now is, again, envision these proteins within this polymer network. With Western blotting, we use antibodies to detect for a specific protein. If our proteins are embedded in this polymer network, these antibodies cannot access these proteins. So we need to actually move these proteins onto something else that will permit them to be detected by a protein. So we now do what's called a transfer step, where you have a membrane. We usually use either nitrocellulose or PVDF, something that is amenable to proteins being running. We're running an electric charge to move the proteins horizontally from the gel onto this membrane. That's why it's called transferring. So you transfer onto a membrane where the proteins are now on the surface of this membrane and are now fully accessible to an antibody to access. And so the transfer step is depicted in the second image from the top, where you have sponges, filters, and you have in the middle your gel and your membrane, and you run an electric field across it. The orientation is important because if you do it the wrong way, you'll just run your sample off the gel onto a sponge paper that does nothing for you. So doing this the right way matters. If you guys do Western blots in the future, you'll remember this moment because I've run it the wrong way and it's no fun. So you want to run the electric field from the gel onto the membrane, and the membrane catches usually most of your protein here. Now you can use an antibody to stain and detect that specific protein of interest. One thing that's important is a key thing to note is that proteins, when you're trying to use an antibody to detect a specific protein, proteins are naturally sticky in some ways. If you've ever cracked an egg on a plate and left that egg on a plate for a while, can anyone remember what happens to that egg on that plate? Has anyone ever tried that before? Clearly, you guys didn't experience enough as kids. But if you leave an egg on a ceramic plate after a while, and you come back to it and you tip the plate over, that egg is just going to get stuck to the plate. So there's a natural, for lack of a better phrase, stickiness to proteins. The reason why that's important is because if you have an antibody that wants to detect a very specific protein, and that's a very specific interaction, you'll have non-specific interactions where there's this natural stickiness of proteins wanting to interact with each other. So, excuse me. So you have what's called a blocking step, where you use some general protein mixture. Usually we use what's called albumin. It's the most abundant protein in our blood. Sometimes research labs, they'll use milk for this step. We'll also use fish gelatin powder. It kind of varies based on what you're doing, but you just want some abundant protein buffer that occupies all of this non-specific binding. So that after you do that step, you can now come in with your antibody that targets your specific protein of interest. And now this non-specific binding is done. You're going to have largely specific binding that happens with this antibody. And so we have our gel that we've run here. We're going to transfer onto a membrane because, again, you can't have their proteins will not be accessible in this gel, in this polymer network to antibodies. So you transfer to a membrane where it will be accessible. On this membrane, you'll block to inhibit any non-specific binding because, again, proteins are kind of naturally sticky in some ways. And you'll, once it's on your membrane, you'll, again, block and stain with an antibody. And usually what ends up happening is we don't want to use just one antibody. Our antibodies, there's a secondary antibody that comes in because what ends up happening is, if you can envision, I'm just going to yell to the computer, I'm not yelling at you guys. In this third image from the top, the primary antibody is binding to your protein of interest. If you had some signal that's tethered to that primary antibody, it's going to be a pretty weak signal. You can amplify that signal by having first a primary antibody and then a secondary antibody that binds all over that primary now, and you have an amplification of signal with the secondary antibody, and then you image the blot, see the color change, and are able to see your specific protein of interest. So primary antibody binds to your specific protein of interest. You come in with a secondary that has some sort of fluorescent or chemoluminescent signal attached to it. And now you can image your blot and see a band emerge for your specific protein of interest. Yes.
I was just curious, like, what if, like, for example, they're trying to write a protein, but then there isn't like that.
Yeah, that's a great question. For the sake of the recording, say you want to do Western blotting, but there's no antibody available for your protein of interest, correct? You will work with a company to make one. So in this lecture, I think we'll probably get to it next class. But I have some information here about how antibodies are basically made, how much it costs if you run into that problem, how much it costs for a company to help you make an antibody for your protein of interest. Trigger warning, it's not cheap. However, by and large, because of how broadly used Western blotting is, as well as antibodies across imaging, across another method called flow cytometry, there are so many methods that rely on antibodies being available for specific proteins that we're hard pressed to find an antibody that is not available for a specific protein. But that's a great question and something to be mindful of, because sometimes the only antibodies available are pretty bad ones in some cases. So it's a great consideration. I'm going to, so we talked about total protein in the first set of slides, where Kamasi blue and lanes 1 and 3 are a mixture of proteins and from some sort of biological sample. And you're able to stain for all of the proteins that way. Excuse me. Lane 2 is a purified sample where there's only one specific protein. But Western blotting, you can load the same samples, whether it's a mixture or a purified version, and you're using antibodies. Again, whenever you hear immuno in the name of some sort of technique, think antibody. And when you think antibody, think specific. This antibody-mediated approach allows you to be incredibly specific, where you can stain for ATPase, as we've all become familiar with in this class, or thyroidoxin, which is another marker. And so you can use Western blotting to look at a specific sample. One thing that's not noted here that you might be thinking is, if you load different amounts in your wells, could that look like there's more protein in this sample than this sample? We usually try and account for that, where you just use an approach that measures the total protein content in your samples, and you load the same amount in every single well. So you load 10 micrograms or 50 micrograms in each well, so that it's even loading. And you'll be able to see between given samples, is there more expression of this protein versus this protein, or this sample versus this sample. And this becomes really important when we start to consider if you take samples from patients that either have some disease or not this disease, and you want to identify, is there some issue with this protein? You can actually identify and see this protein is truncated, meaning it's not fully expressed and it's only a short part of it, because that protein that you're probing for some reason, it's supposed to be here. You know it's supposed to be here. It's showing up down here. So there's lots of diseases where proteins are truncated because of some mutation that stops their expression at a certain point. And you can actually run a Western blot and see that it's an altered truncated form. So this actually becomes really helpful when trying to characterize proteins that play a central role in disease. I'm going to stop there and we'll pick back up where on this slide next week. Sorry again that we had to do only because if I keep going, I'm going to eat into a lot of the slides for next class. But and I also usually run late, so I thought it'd be nice to end a little early and give you guys some time. But we're going to pick back up here next class. Thanks for your time.